Hereby I open this academic ceremony in which Mr. Muhammad Ali will defend his academic thesis, Integrative Network-Based Approaches for Modeling Human Disease. I would like to invite Mr. Ali to give us a presentation and a summary of the work you have been doing to write your thesis, and that within approximately 15 minutes.
Dear Rector, thank you very much. Dear Corona members and colleagues, thank you very much for coming. Today, I am going to present the computational work that I carried during the course of my PhD entitled Integrative Network-Based Approaches for Modeling Human Disease. As my work is based on the modeling of human diseases, let's start from the basics of human biology. As we know that we as humans has many organs that do specific functions in our body. For example, brain and heart do different kinds of functions in our body. These organs are essentially made up of tissues, which are a collection of cells, similar cells, that all carry the genetic information in the form of DNA. This DNA can be further subdivided into functional units called genes. Although all different types of cells in our body perform different kind of functions and they look different, but the underlying genome sequence that they carry is essentially the same. So the differences in their structure as well as in their function arises from the epigenetics, which decides which sequence should be allowed to be used by a cell and which shouldn't be. So the differences in them actually comes from the epigenome that sits on the top of the genome. And the specific characteristics of this epigenetics, such as different epigenetics markers, such as DNA methylation, as well as the histone modifications, make certain parts of the DNA accessible to the cells so that they can use this information to carry out their function. As we develop from one cell to the trillions of cells, the epigenetic markers also keep on changing through the entire lifespan of a human being. So they keep on changing with time. Also, another important factors which can affect these epigenetic signatures is the environmental factors such as stress or the physical or mental activity. So now you can think about that maybe the diseases also carry their particular epigenetic marks which are different from the healthy individual, and that indeed is the case. For example, in Alzheimer's disease, which is characterized by the accumulation of proteins called tangles and plaques in the brain cells, physically we see a reduction in the size of the brain. And if we analyze the genomic data that we get from the disease as well as from healthy people, then we realize that there are certain epigenetic changes as well as transcriptomics changes that are unique to the disease patients. So keeping these things in mind, the main aim of my thesis, of the projects that I did, was first that in order to cure these diseases or halt the progression of these diseases, one approach could be to come up with the cellular conversion. So we know that in some of the diseases, for example, in Alzheimer's disease, certain neurons, they degenerate, and in many other diseases, the cells start to malfunction. So there, if we could convert the cell types, those desired cell types which are depleting, then we could cure those diseases. So the first aim of my study was to predict the transcription factors that can help us achieve desired cellular conversions. And the second was to identify the differences in the disease as compared to in the healthy individuals by using computational disease modeling. Beside the epigenetic regulation, another important layer which plays a crucial role is the transcription regulation. Although it is a very complicated process, but I would try to explain it by the help of a toy model here. Let's consider 3 genes which are transcribed into messenger RNAs and subsequently to their protein. Now, as we know that the complex systems, biological systems, are made up of different components, which are in this case genes, and they interact with each other to carry out the normal functions of the cell. So in order to understand their function, their mechanism, we need to understand how they interact with each other. This information can be, we can get it from the literature or from the databases. So for example, here, like the product of gene 1, which is protein 1, regulates gene 2, and then product of gene 2 regulates gene 3, and so on. This information can also be represented in a more easy way in the form of a gene regulatory network, where all these genes are connected to each other based on their regulatory functions. Now we can also overlay data from different omics layers onto these GRN models to make them more accurate. For example, we can overlay information about the DNA6, the chromatin accessibility information, which regions are open and which regions of DNA are open and which are closed. Also active enhancers data and active promoter data. So using this, we will be able to find that which interactions make sense according to both different layers of regulation, and hence remove the interactions which are not compatible to them. Overall, in my thesis, I defined 4 main projects on which I worked. First of them was integrating data from different layers, specifically transcriptomics and epigenetics, in order to identify, to build the core transcription regulatory networks, and then use those networks for predicting the transcription factor that can help us induce a desired cellular transition. In a second project, I integrated data from different omics layers, named genomics, epigenomics, and transcriptomics, in order to prioritize the AD-associated genes. Then we increased the resolution of our analysis and focused on only the genes that belongs to a specific pathway related to lipid, and see what are the genes and pathways that are compromised in this disorder. Last but not the least, I use a differential GRN-based approach for modeling an early neurodevelopmental disorder called Barton disease. Due to the shortage of time, I would go in detail of the only first 2 projects. So in the first study, as explained earlier, we use data from epigenetic and transcriptomics layers, and we use that information to build core transcriptional regulatory networks for different cell types. By using those core networks and the differences in the epigenetic and transcriptomics landscapes, we were able to predict transcription factors that can help us in inducing the desired cellular conversions. So before we go into the details, why we needed to come up with this method is basically because it was in 2006 that somatic cells from the patients were converted into iPSCs. These induced pluripotent stem cells has the potential to be differentiated into any desired cell type of our interest, which made it possible to model the human diseases in the laboratory. So here, for example, in order to generate model for neurodevelopmental disorders, we can obtain somatic cells from the patient, convert them into iPSCs, and then we can further guide them towards the neural fate. Then further upon differentiation, we will get different neural precursor cells, which will be further maturated into different kind of neurons, or different kind of brain cells that can have many different applications. One of them is serving as a disease in a dish model. So the recent studies suggest us that instead of going through all this way, which is time consuming as well as laborious, there are techniques by using transcription factors and microRNAs. We can directly convert iPS cells into any more differentiated cell type that we want by a process called directed differentiation. Also, the matured cells can be interconverted into each other by using a specific recipe through a process called transdifferentiation. All these processes carry a common limitation, which is the cellular conversion efficiency, the number of cells that are converted when you use the transcription factors or any other medium to conduct the directed differentiation or transdifferentiation. In order to overcome this bridge in the this gap in the literature, we came up with this InteragNet method, a computational method, where we integrated the information about the epigenetic marks, DNA's accessibility, with the Booleanized gene expression data in order to reconstruct the core transcription regulator network, and then using the differences in the epigenetic landscape as well as in the transcription of the genes, we identified the set of transcription factors which can help us achieve any desired cellular transition. Thus, we also experimentally validated this method in the laboratory, where we wanted to convert iPS cells into human memory epithelial cells by a combination of different transcription factors. The preliminary results that we obtained were convincing and promising, such that a lot of cells were converted into the desired cell type as compared to the negative controls. Thus, we can conclude that the approach that we have developed can help us in building core cell type specific networks for different cell types, and by using those core networks, we can achieve desired cellular transitions from any starting cell type into a final test type by using the minimal set of instructive factors that are required for that purpose. In the second study, I integrated data from different omic modalities, genomics, epigenomics, and transcriptomics, to prioritize the Alzheimer's disease associated genes based on their mediator effect, which means that which are the variations in the genome that the genes that have been already susceptible to AD also have a causal functionality. So in order to achieve that, we integrated data from genomics, epigenomics, and transcriptomics onto a functional gene-gene interaction network, and then use the network diffusion algorithm, a computational algorithm, for prioritizing the genes. Using this approach, we were able to rank genes based on their effects, based on their importance. Then we extracted the subnetworks of these top-ranked genes to identify which are the other genes that are connected to these highly ranked genes, how they interact with each other, and then use their gene expression signatures to identify existing drugs that can help us in reverting their gene expression program. Thus, we can conclude that by integrating data from different layers of omics in a systematic fashion, we are able to rank the genes based on their importance in of being dysregulated in the disease, and also understand the underlying mechanism behind how this dysregulation happens. Furthermore, we are able to predict the active drugs, which are existing drugs that can help us in reverting the gene expression program from healthy towards from disease towards the healthy phenotype. Overall, the work conducted in this thesis, and which is also written down in this thesis, suggests us that integrating data in a more systematic manner help us in getting the core TRN as well as genome-wide gene regulatory networks, which can help us in understanding the differences between the healthy and the disease phenotypes, what are the genes that are involved in and maintains those phenotypes. And this kind of information can help us in ranking those genes as well as knowing the underlying mechanism behind their dysregulation, which enable us to predict the drugs or come up with novel intervention strategies. These GRN models, when built for a specific pathway, such as in this sphingolipid, then followed by a network perturbation analysis, we can identify the genes that that contributes towards the AD pathology. Moreover, the differential GRN inference approach, which was used in Alzheimer's disease, where we get two networks, one representing the disease phenotype and the other the healthy phenotype. By using those network and their topological analysis, we were able to identify genes and pathways that are compromised in this early neurodevelopmental disorder. Thank you very much for your attention.
Thank you very much for your clear presentation. We start now the opposition, which will be opened by Professor Prikkaert, Professor of Experimental Neuropsychopharmacology at our university.
Thank you, Mr. Prorector. Dear candidate, first of all, I would like to congratulate you with your impressive thesis. And I also would like to congratulate your promotion team. It's a big one, who I know very well, and they are real experts and pioneers in this exciting and emerging field of research. It's impressive work because it entails combining huge data sets of databases and measurements of ex vivo and in vitro epigenetics and RNA sequencing, so it's a lot. I'm a neurobiologist slash pharmacologist, so I learned a lot. It was difficult to read for me, by the way. I do hope that your work will also be published, because I noticed only a book chapter is published yet, and that would give other researchers the opportunity to also learn from your exciting work. Since my personal interest is in pharmacology, I was extremely interested in your chapter 4, as you might imagine. So, if I got it right, you performed a gene regulatory network approach to find new Alzheimer targets for old drugs. This is called repurposing, and this is something I'm also doing, but I'm doing it old school, I would say. But so your conclusion is that a combination of cycloserine and apramycin would be the most effective therapy treatment for AD. So my first question is very simple. Do you know for which diseases these drugs are on the market, actually?
Highly esteemed opponent, thank you very much for the compliments and an interesting question. So about these drugs, the existing literature suggests us that apramycin, cycloserine has been already used to increase the cognition system in the Alzheimer's disease patients. So that drug has already been, I guess, predicted for this purpose, whereas for the apramycin, we do not find any existing literature suggesting that it has been used, but the literature still suggests us that it helps us in the maintenance of the, if I'm not wrong, the maintenance of the cells, or also the maintenance of the cytoskeletal muscle cells. So these kind of, as they are playing some role from the maintenance, then from the AD we know that this is a neurodegenerative disease where neurons degenerate, so maybe it could help in making the cells or neurons more stable. And that's...
Okay, yeah, you start already speculating about the mechanism, but I actually ask you for which they are on the market. And cycloserine is not on the market for Alzheimer. It has been tested for Alzheimer, like you referred to in the paper, but these are very old studies from the previous century, actually. But both drugs are on the market as antibiotics, and especially cycloserine is on the market for tuberculosis. And you nowhere mentioned the word antibiotics. So anyway, but I was very interested, so I checked the whole list of drugs for what they are on the market. So, so if you can go to table 4 on page 61, so I will go quickly through them. Azoxycan, difficult to pronounce it, is a non-steroid anti-inflammatory drug, so anti-inflammation. Ginkgolide A is made from ginkgo biloba leaves. That has a very broad action. So it also affects PD5, which is the mechanism of Viagra, actually, but mostly cerebrocardiovascular. Then we go to gentamicin, antibiotic, ondansetron, anti-emetic against vomiting, but also has anti-inflammatory action. That's maybe why it links to the gene you report, linked to it, IL-15. Thioguanosine, anti-cancer, cefuroxime, antibiotic, then CP944629, anti-inflammatory, and chlorambucil, anti-cancer. So many antibiotics, anti-cancer, and anti-inflammatory. This might tell you something. So my question is, did you notice this? If so, what does it mean for Alzheimer's disease then?
So the one of the drug that you mentioned having anti-inflammatory effect, that we also can think about associated to Alzheimer's disease, as the inflammation is one process that we observe. Other than that, why we are getting, let's say, more antibiotic drugs is the database that we are relying on. So for this study, we relied on connectivity map database, which actually profile these drugs on different cancerous cell types. And that is, to my knowledge, the most comprehensive resource at the moment, which is effect of these drugs on different cell types. So by using our gene expression pattern that we get, the signatures, we are interested in finding the drug that could revert them. So that's why we go to this database and then look up for the drugs that produce the required gene expression, like which is the anti to the disease gene expression signature. So if the drugs there are not related to Alzheimer's disease, are more into antibiotics or so on, it gives us an idea that maybe they could be also used for some other purpose, like in the AD. So I would not rule out the effect that if they are antibiotic, then they could not be a potential drug for Alzheimer's disease, because here the aim was to revert the gene expression program, and if these drugs hold this power to do that, then I guess they could be good candidates.
I agree in a way, but I still have the feeling you do not check the drugs properly enough. Also mechanistically, although you have in the first answer already tried to link it more to the mechanism, because I'm also checking the mechanisms of these drugs. And there's actually one drug I found the most interesting one, or that's ondansetron, antiemetic drug. Do you know the mechanism of action of this drug? Because I also find it important, if you mention drugs or mention genes, that you check the mechanism. Did you check that mechanism? Do you know what it is? It's not bad if you don't know, but just out of curiosity, if you know it.
No, we actually didn't check the actual mechanism of these drugs, because it could be very complicated to go into the mechanism, which are, I also believe that they are not very well studied and clear to understand how these drugs affect their mechanistic. So
ondansetron is studied a lot, because it's a 5-HT3 receptor antagonist for a serotonergic receptor. And that's actually very interesting for Alzheimer's research, because if you would do research, you will find that until recently, alpha-7 nicotinic receptors have been heavily investigated for Alzheimer's disease. And they didn't make it because of adverse side effects, unfortunately, on the GI tract. But these alpha-7 agonists also target the 5-HT3 receptor as an antagonist. And actually, some people think that's actually causing the cognitive improvement. So I see this target now popping up again, and I worked a lot with the pharmaceutical industry on this. So I think it's, based on your data, it helps me. So what I would like to say is that in the future, that you check your author list, you should also include a pharmacologist.
Definitely.
When you start talking about drugs. And this, we can join forces and come up with a new, interesting, strong target.
Sure.
What is your opinion on that?
I really agree with what you just suggested, because as I'm from more computational background, so going into the mechanistics of these drugs and more biology is not my area of expertise. So in this case, if we could collaborate, as also this is just a chapter thesis at the moment, and still we are polishing this work. So I guess if we collaborate with some expert in this pharmacology like you, then it would really increase the impact of this study.
Does it have to be me? But thank you very much for your kind answer. Give the word back to the prorector.
Thank you very much. We now have 5 opponents left. 4 of those are coming from other universities, and I would like to thank them collectively. Normally I thank each participant in the corona individually, but there are so many now, and it would take so much time to thank them all, that I do that collectively. And the first one is coming from, they are coming from 4 different countries, Luxembourg, Belgium, Germany, and the Netherlands. So the first one is coming from Luxembourg, and that is Professor Schwamborn, Professor in Cellular and Developmental Biology.
So, dear Mr. Prorector, thank you for the introduction. Dear candidate, thanks for the kind presentation and the thesis. As you might imagine, my question go more in the biology direction. So within the thesis as well as your presentation, you mentioned stem cells and progenitor cells. So how would you define those two?
Highly esteemed opponent, thank you very much for the compliments and the question. The stem cells, I think they are more plastic in their nature as compared to in the progenitor cells, because progenitor cells are specific to one fate. For example, neuroprogenitors or blood progenitors and so on. They can further differentiate only into, if they are neural progenitors, then into neural cell types, blood progenitors into blood cell types. But in contrast to that, iPSC, they hold the potential to differentiate into any of these progenitors. So to sum it up, I would say they are more plastic in nature. You can direct them in any fate that you want as compared to the progenitor, because they are more primed already towards particular fate.
That's probably okay for distinguishing the two, but what would be the true definition of a stem cell? So what does a cell have to be able to do to be called a stem cell?
To make its own copies. So if there is one stem cell holds the potential that they can renew themselves. So I guess that's
one of the two criteria. What would be the second? So what you say is correct. Self-renewal. This is one of the key characteristics. There's a second one.
Maybe self-maintenance as well?
No, that's pretty much the same. It's more the ability to differentiate. Because otherwise the cells would basically just be able to proliferate and become more of itself, but not differentiating. So these two.
Sure.
Okay, then if we move to the Batten's disease chapter, what's happening during Batten's disease? What the characteristics of the disease?
So this is this is actually a disease that happens in kids in early age. So with the passage of the time in the first decade or the early second decade, you see a loss in the vision, as well as the degeneration and inability to understand and perform the task completely, and premature death in the late second decade or early third decade of the life. That is the physical characteristic hallmark of the disease.
Yeah, and can you describe just briefly the gene expression analysis that you did there?
Yes, so we had actually the RNA-seq data from the different the brain organoids that belong to the Batten disease model, because that is actually a model of to study the effect of a single gene mutation, which is C2T at 1054 base pair in the CLN3 gene. So we had this mutant cell type which represent Batten disease and the control cell type which represent healthy. So we had the RNA-seq data from these tissues, the brain organoids, and there I conducted the uniform processing pipeline using the top hat 2 tools to perform the differential gene expression analysis.
Okay, and then you have list of differential expressed genes.
So once we got the differential gene expression, we got a list of, let's say, 1,000 genes. We are not we don't know what are important ones and what are not. So for that purpose, and also to see what are the changes that are happening in this disease at a system level, we opt for the gene regulatory network approach. So there we connect all these differentially expressed genes using the existing literature. And then we conduct, so then we get a phenotype, a network which represent disease phenotype, and the other representing the healthy phenotype. And then we use the disease network for network simulation analysis, in which we perturb different genes in the network and see how do that effect propagate in the network, which gives us an idea about the stability or maintenance role of those genes in the network. And they could be the potential target for designing novel intervention strategies as well.
That would have been is exactly my follow-up question there. When I now have these targets, how do I use that to come up with a treatment strategy? Because that's ultimately what you want to do, or you want to do.
Yes, so once we identified these key genes, and as per literature, we know that yes, they play some important roles, then we should look into the expression, whether they are upregulated or they are downregulated. So as we are in a differential expression setting, which means that if a key gene which is upregulated in disease is downregulated in healthy phenotype, we want to reduce its expression. So the thing that we should look forward into maybe a knockdown study, that we knock down that transcription factor or gene and see what are its effect on the other genes on the other genes, whether the cells move more towards the healthy phenotype by doing that kind of knockout, or what else. So,
but this is more like a follow-up experiment that you would do in the lab. How would you use that therapeutically? So what would be the therapy approach?
So if, let's say, if this works in the lab, then we know that we have a target, which is a drug, which is a gene, and we want to downregulate it. So then we will go from the pharmacological point of view, that what are the drugs that can help us in downregulating this gene. So that are, let's say, a blocker of that gene, or they bind the receptor of that gene so that it does not transcribe or translate, so that there is no product of this gene into the cell, in the cell. So we should look for that kind of drug. Then.
Okay, thanks for the answers, and thereby I give the word back.
Thank you very much. The next opponent is Dr. Van Mierlo from the Biomedical Research Institute in Hasselt University, Belgium.
Thank you, Mr. Co-Rector. Dear candidate, first of all, I'd like to congratulate you with this excellent thesis, also the promotion team, promoters and co-promoters. I think it's well written and I learned a lot. As a bioinformatician, you studied the integrative network approaches in different human diseases. And I think there's a big need for this to couple the multiple layers into a combined analysis. So I was impressed by the clear way you explained this in your thesis. Nevertheless, I'd like to challenge you with some questions. So my first question relates to chapter 2, and it's on the use of iPSCs derived from patients. It's a review you write there. And I'd like to discuss with you what your view is on the one level. You have the iPSCs, which are stem cells. Which are stem cells, which you describe multiple layers, and on the other hand you have tissue of patients, in which you also have these different layers. But when you look in sporadic diseases in which you have a lot of epigenetic influence, the environment which is inducing these epigenetic influences, they might have effects on for instance, brain cells in diseases like Alzheimer's disease, but also multiple sclerosis. But if I then look at iPSCs, for instance, iPSCs from oligodendrocytes, which play a crucial role in the myelinating in multiple sclerosis, and which are particularly hampered to do this myelination. When you look at iPSCs derived from patients who cannot remyelinate anymore, these iPSCs, they can myelinate. So clearly there's some difference on the epigenetic level. So according to you, how can we translate the findings from epigenetic layer in the brain of Alzheimer's patients or MS patients to iPSCs, and how should we interpret these data?
Esteemed opponent, thank you very much for the compliments and the question. If I understood correctly your question, then you are asking that the iPSCs that we get from the MS patient, the epigenetic marks that we get there are different from, sorry?
From the marks which you would find when you analyze brain tissue.
From another brain tissue or from another person?
Yes.
Patient, so on. So you are asking what would be the differences between them?
Yeah, and what will be the implication for the interpretation of experiments which you do on these iPSCs?
So here there are two ways to look at this information. For example, in the case of Barton disease, we use the iPS cells and then inserted the mutation because we didn't want to also consider the genetic makeup of the patient cells. So the other approaches or the existing approaches, what they were doing was obtaining the cells from the patients and then converting iPSCs and the Barton disease model. But there we know that those cells are from patients and patients have their own genetic makeup. And we were only interested in studying the functional effect on that one mutation that I mentioned previously. So similarly, in the case of other diseases, if you take a sample from a patient, then definitely beyond the epigenetic marks which are associated to those diseases, there is a certain epigenetic genetic makeup of that organism as well, which due to disease has been changing. And if it is a mutational disease, for example, in the Barton disease, in the CLN3, then we can incorporate that particular mutation and just study the effect of that, the functional effect of that mutation. So this, I mean, it helps. It depends on the study, the question that you are asking. If you are interested in knowing overall if changes in a disease versus a healthy person, then you can go this way. But if you are interested in knowing only the effect of one particular mutation or a change in the expression of a gene or so on, then we could go for the other approach to insert that particular mutation by CRISPR-Cas and then study its effect.
Yeah. And also related to that, if you like to do a drug screening, would this have consequences? If you have an iPSC which is epigenetically reshuffled?
So the drugs for the iPSCs, you mean?
If you like to test, for instance, a pro-myelinating substance, and you're testing it in an iPSC derived from a patient, but the epigenetic imprinting, which is actually involved in causing the disability, is not there anymore in the iPSC. How will this affect your interpretation of potential results on iPSCs?
I think then in this case, maybe a good idea would be if we could also genetically alter the iPSC's signatures in such a way. Maybe I'm not sure if CRISPR-Cas can do that. That we could also add these signatures that could help to iPSCs to also myelinate. If they can do that, then they show all the physical the hallmarks of that disease that we want, and then we can do the drug screening and identify which drug can help neutralize these signatures.
Okay, thank you. I have an additional question, and it's more relates to chapter 3, to the Integrative, which you set up. But it also relates to the iPSCs, because in that chapter you identify a lot of factors, transcription factors, instructive ones, but also core transcription factors, which are crucial to differentiate iPSCs into their final phenotype which you like to study, right? Yes. Still, even though you identify a lot of these core transcription factors and instructive factors, the efficiency of differentiation remains rather low. So what might be the cause here, according to you? And might there be a role for the timing of the expression of transcription factors in iPSCs?
Yes, that's an interesting question. So there are two ways. We are not just going from iPSCs to the differentiated cell types, but also the way back, because we also consider them as cellular transitions. So we can predict the combination for both of these cases. Now, coming to the question, why still the efficiency is limited, it actually goes back to the epigenetics layer as well. So it is not only the transcriptional layer which is involved in this case, but also the epigenetics. And that was the thing that existing studies were only relying on the transcriptional information. What are the changes in the initial and starting cell type? What are the genes which are very lowly expressed in starting and very highly expressed in the destination cell type? You upregulate in the starting and somehow you end up there. But they do not take into account the epigenetic changes, like the chromatin.
I agree on the epigenetic imprinting. I think this is a very valid point here. But where I try to go to is the timing of the expression of transcription factors in those iPSCs might be crucial for the efficiency of the differentiation. And is this something which you could take into account into your model on time?
In the current model, it is not incorporated, but definitely this is a valid extension of this method. For example, if we are going for a directed differentiation between more mature cell type, then there exist also approaches where first mature cells make more plastic towards the iPSCs and then differentiated towards the final cell type to first make them more kind of vulnerable so that they can move, they can go in another direction, and then applying the transcription factor that can help to convert them into that target cell type that we want. So definitely the timing of the transcription factors also play a very important role, but this thing was not considered here in this model.
Okay, thank you very much. I'll give back the word to the Prof.
Thank you very much. The timing plays also an important role during the defense. So we move on to Dr. Ramirez, head of the Division of Neurogenetics and Molecular Psychiatry from the University of Cologne in Germany.
Thank you, dear Prorector, dear candidate. I would like to thank you for the nice presentation, also for your thesis and also the promotion group. And in sake of time, I would like to go directly to the questions. And I would like to focus on the chapter 4, also because I think that the questions I have might change a little bit, perhaps your results. Because, as you know, genetics and Alzheimer's disease have been growing, and with these new high-throughput technologies, we have found a lot of different genes and different approaches have been used, including pathway analysis. And now you're proposing a new approach that combines different levels, and which is a very valid and it could be a very interesting approach. And you started using genetics, although you mostly focused on epigenetics and transcriptomics. Your starting point was genetics, and there you use a summary statistic of the IGAP consortia, and then you select 11,000 genes that you say these are the and those 11,000 actually gave results to 1,500 genes that you call AD genes. So I would like to ask you first, why you call this AD genes actually?
Esteemed opponent, thank you very much for the compliments and the question. So the first starting point here is the genes which are already susceptible for the AD according to the IGAP study. And then we, based on the GWAS analysis, we get the level of significance if those genetic variants are significantly associated to Alzheimer's disease or not. So there, by applying the thresholds, we filter out for a set of genes which are significantly associated to Alzheimer's disease based on the p-values. So it is not actually AD-associated AD genes, but AD-associated genes, and which is our starting point.
So you said that these 11,000 SNPs that you took are all associated with AD?
Yes.
Because if you think about GWAS, actually there is a threshold for genome-wide significance. You know what is the threshold?
I think it is 3 into 10 to the raised power minus 8.
5 times 10 to the minus 8, exactly.
Yes.
And this means that these genes, so if they cross this, you consider these genes actually associated. Most of the genes probably are, or these associations are spurious associations. And then you assign this, so let's say we have this 11,000 and they probably are those true. So we go and assume that is true. And then you assign those SNPs to particular genes. And how you did that, actually? Why you decide that which gene is actually the one that it's the right one for that SNP?
So as you mentioned that 11,000 is the starting point, and yes, indeed, they are not reaching the genome-wide significance. So, but we have a hunch that they are associated to AD, but then we have also data from different layers, epigenetics and transcriptomics, to take a look into those genes, whether there are changes in the epigenetics or in transcriptomics of these genes or not. So that's why it's better to be a bit relaxed at your starting point. So once we are there, when I associate the genes, the probes, I took for the most significant one. So for every gene, you get multiple probes, which are differentially methylated, but there exist different criteria. Some people actually use the mean, some people use the median of this. But in our case, we wanted to consider the
because you are moving now to the epigenetics. I want to stay into the genetics. So the definition of your genes.
Mm-hmm.
So why you say the gene that is the SNP is beside of the gene, this is the gene. Why? What happened there? Why you use this strategy? I mean, there's other ways of doing that. And this is one of the major issues actually in genomics.
Mm-hmm. So what we did that to annotate the SNPs to the genes was to use the Homer annotations. So that is a tool which is used for annotating the peaks as well. So here we consider the SNP site as one base pair peak, and then we annotate it to the nearest gene. So by using the Homer tool, I annotate every SNP to the gene that is most closest to, and then we consider the unique genes that we get from there.
So this means that you will, if we do follow that strategy, so I'm surprised that actually you didn't found any pathways related to immunity and cholesterol metabolism, lipids metabolism. So why is that? So why all the genes you have in your list are actually related to cancer, ETS1, Wilhelm tumors, and these genes, if they are so relevant, because you rank them very high, why they were not found in the GWAS? Because genetics, I'm geneticist, so I'm biased. Why these genes were not found in the GWAS? Because genetics is sometimes before epigenetics, because you get it when your sperm and the oocytes get together. So then you have before any disease process start. Why is that?
You're asking that why these genes are not significantly associated with
why you do not find the things that other approaches using genomics, you're finding completely different results using a very small and limited data set.
Mm-hmm.
And I think that's what I want that you tell me why you think that it could happen.
So I would say that these genes are already associated as per the GWAS analysis, because that is our starting point, 11,000, and ETS1 is among them.
That's not correct, because if you look at the results of the GWAS, if you look at the Manhattan plots, even the last GWAS published this year, there's none of these genes on those top hits. Mm-hmm. So clustering, APP is also not there. These are one of your top hits. So I think you missed probably something that is the LD block. So naming the genes just because of this nearest one, I think it is as a problem, because you forgot to use the entire locus that could be including other genes. So you have candidate genes, and that is one of the issues of the GWAS, that you target an LD block which might contain many different genes. Mm-hmm. So assigning just to the one gene that is beside is probably a mistake that can produce bias in your analysis, and this might have produced the results you have. Would you agree with me on that?
I think now, if you are going for this, I find this as a valid approach. We see that the genomic interactions are happening even from different chromosomes, so we cannot really do that assign every SNP to the closest genes. But this is, I would say, one of the widely known approach to do that in that way, because we cannot take into account the 3D organization of the chromatin by using high-seq data or so on. So the most, I think the most reasonable assumption here to associate it to the nearest gene makes kind of sense.
Well, I think on the sake of time, I will just give it back, but I do not agree with that, because there is EQTLs that shows that sometimes the expression of a gene does not be necessarily beside the gene. Sometimes you have 3 kb, so how is the limit you say what is nearest, you know?
Yeah. That's same happens for the enhancer annotation as well.
Exactly, so I think that's a point, you know, so just the nearest gene might be a problem to use that strategy.
That I agree.
Go back to you, Prorector.
Thank you very much. The next opponent is Professor Martinez, Professor of Neuroinflammation of Neuropsychiatric Disorders at our university.
Thank you, Mr. Prorector. I would like to congratulate the candidate and the promotion team for a very nice thesis and a lot of work in short time. My questions, as you might expect, will go to chapter 5, because it's my area of expertise on the lipids. And there I would like you to look in page 79, because, give you the time. So you use the in silico network perturbations to identify the most influential combinations of genes in the gene regulatory network, representing the AD phenotype. So in the table 7, the first 3 gene combinations are presented by TNF, interleukin 2, and MAP kinase 3. None of them result to be significantly differentially expressed in your previous analysis. So could you explain why?
Hi,
yes.
Highly esteemed opponent, thank you very much for the compliments and the question. So coming to whether they are why they are not significantly differentially expressed, the first starting point here was that we identified these sphingolipid associated genes, which were 252 that we could found, and then we look for their differential expression. Why they were not significantly differentially expressed might be due to the limited number of sample size that we had. We had actually 82 samples, 46 AD and 38 for the controls. And this might be the one reason that they are not, but why they are ranked higher here goes back to their importance in the network. Although they were nominally significant, but in the network they were the key genes or the hub genes, like they were regulating the expression of many other genes in the network. Most probably also the significantly differentially expressed will be downstream of these genes. Like if you can target these, you can also target those ones.
So, and why do you think is that? Why do you think they have such a big effect on to revert the expression level of 41 genes? So what can be the reason for that?
I think it's a valid point that sometimes the changes in the gene expression are subtle, but their effect is more strong. For example, the upstream gene might not be differentially expressed due to, there could be many reasons for that. It is that their expression is not significantly differentially expressed between disease and the controls, but they can play an important role in regulating their downstream genes. Mm-hmm. So in this case, indeed that is the case, that they are not significantly differentially expressed, but they are able to regulate many other genes which are significantly.
Okay, so in your analysis, you use a area of the brain that is the middle temporal gyrus. In Alzheimer's disease, the hippocampus is mostly affected. So my question is, why did you use that region? And will you expect differences if you will have analyzed other brain regions? Do you think this is robust or not?
I think if we go for the other brain regions, for example, prefrontal cortex or the one you mentioned, definitely we will find some other different results than this, because different brain regions are affected by AD in different stages. So we cannot say that all the expression of all the genes in all the brain samples, if we extract samples from different brain region, would be similar. It would definitely be different. In this study, why we focused on this was because of the consortium study that we have. We only got the data for the MTG and also the prefrontal cortex. So this could be a nice extension of this work, that if we further want to see if a sphingolipid mechanism is really disturbed in only certain brain region or in all of the brain regions, then definitely this is worth going into that direction.
So if that's the case, so why do you think, or how do you think your analysis and your study can help to a therapeutic outcome or can go to the clinic? If you think that there are differences between different brain regions, how will you envisage that you can bring this to the clinic? What do you need to do then?
Let's consider that it starts from the middle temporal gyrus. Then in that case, if we are targeting that in that region, then we do not need to worry about what will happen in the prefrontal cortex or in the dorsal raphe nucleus. So in this case, if we are able to revert the gene expression in one particular brain region, then definitely it will have effect on the other brain region as well. As in the progression of this disease, the brain regions start to get the neurodegeneration happens subsequently, not all together in all the brain regions. So if you, let's say, cure at the stage 1 in the brain, in the MTG region, then maybe it will have its effect translated to other brain regions in time, with the passage of time.
It could be that the effect in other brain regions is not adequate, right? So you target one brain region. How will you do that? How will you target that? So do you have any idea on how you could bring this further to a next step?
Yes. So here, as we identify the underlying mechanism from which we mean that which was the effect of the methylation at that gene. For example, if we consider these 3 genes, the top 1 perturbation candidates, then we see what are their changes in the methylation. Let's say they are all hypermethylated, which means that all these genes are downregulated. We also see these in the gene expression level, that all these genes are significantly downregulated as compared to the healthy person. So there we get this idea that maybe this CPG sites belonging to these genes are functional. If we can epigenetically modify them, do the epigenetic modification by CRISPR-Cas, remove that hypermethylation, let's say with hypomethylation, then it might upregulate those genes, hence reverting the gene expression program from disease towards the healthy.
Okay, thank you, esteemed candidate, and I give the word to the prorector back. Thank you.
Thank you very much. The next opponent is Dr. Glaab from the Luxembourg Center for Systems Biomedicine at the University of Luxembourg.
Thank you, Mr. Prorector. Dear candidates, one of the key new aspects of your work is that you integrate multiple types of omic data to find key disease-associated network perturbations. So I was wondering, how would you use bioinformatics approaches and potentially further biological data sources to further characterize these network changes in terms of whether they are more likely pathological or protective and more likely causative or non-causative?
Esteemed opponent, thank you very much for your compliments and the question. So actually there exists databases like that, for example NeuroProDB, which contains a list of genes that are neuroprotective genes. So in this case, if we identify the genes which are, let's say, the key genes here, then we could go into this database or literature search to identify whether a gene is having a neuroprotective role or a neuropathological role. So then if a neuroprotective gene is downregulated, then we need to upregulate that so that it could perform its neuroprotective function. Similarly, if a gene is neuropathologic and it is upregulated, we need to downregulate that gene in order to minimize its pathology or its contribution to the pathology. So then we would like to downregulate that gene. So such a way we can classify our genes that we get, the key candidates, into neuropathologic or neuroprotective, and then based on their gene expression signatures and their classes, neuroprotective or neuropathologic, we can then look for the desired gene expression program that could be reverted.
And can you also use bioinformatics approaches to get an estimate of whether a change is more likely causative or secondary, so rather downstream change?
Yes, indeed, that is also done in this case, that when we start, we are not sure about these genes, whether, for example, in the fourth chapter, the AD signature prioritization. So in the beginning, we are not sure about these AD susceptible genes, whether they are causative or not. Then we look up into the methylation, the epigenetic layer, then transcriptomics layer, and then we also, using network topology, identify how the dysregulation in this gene resulted into the dysregulation of many other genes in the network. So now, once we have characterized that chain, which are the changes here, and how they can help us in reverting, then we can devise the exact strategy for that. So in short, the analysis conducted in this particular chapter guides us also towards whether the genes play a causal role or not.
A question on a different topic. I noticed that in your thesis, you mostly focus on human biosamples. However, in public data services or public databases, there's also much data available on animal models for the specific diseases that you focused on. So could you think of some approaches to integrate also this animal model data on a network level with the human biosample data that you analyzed, and how could you use maybe orthology relations between animal model genes and human genes to build such a combined human-animal model network analysis?
Definitely, this could be an extension of this work, and also it gives more confidence if we find the orthologous gene and the same hits appear also in, let's say, a mouse PD model or a mouse AD model, then it will give us more confidence that the genes that we have highlighted really play a key role across different organisms as well. So they are the key genes in this case. But why I was restricted to the human was also the application of this part, which was the eventual goal is the therapeutic intervention. So in that case, we were more interested in doing those analyses in the humans, so that if we can find the collaborators which can help us also validating those experimentally those predictions.
And how would you go specifically about human-animal model network analysis? Would you build a combined network that contains both human and animal model data, or would you build two separate networks, one for the human species and one for the animal model species, and then compare them? So what do you think would be the most sensible and most useful approach here?
I think I will keep these things separate, not mixing with both, because I believe that not all the human genes can be translated or we cannot find orthologs for all the genes. So there will be many similarities, but also a few differences. So it's better to keep the things separate, but compare the hits that you get from one organism with the hits that you get from the other organism and see if they overlap. If they overlap, that gives you an idea about the power of your approach. And if not, then as these are different organisms, then definitely you expect also, aside similarities, some differences.
And if there's still time left, maybe one last question. So you also speak about potential applications in biomarker modeling. So I was wondering, how could you move basically from your network analysis to a machine learning analysis that also gives you predictive information about the disease?
So here we can use different machine learning algorithms. For example, we can use random forest. We have the data, and then we want to, let's say, identify which are the signatures, epigenetic signature that correspond to the disease, and the other correspond to the healthy. So we want to classify them.
You may briefly finish your answer.
So, as it is, if we take it as a classification problem, then definitely machine learning algorithm can help us in this approach. For example, even also for classifying if a data belongs to a patient or healthy, then the machine learnings help us in do the classification.
So, Mr. Ali, thank you very much. You will have noticed that the time appointed for defending your thesis has passed. The degree committee will now withdraw, and we will discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations in this room, and we will return in this room in a couple of minutes. Thank you.
Thank you.
Congratulations. You can go to family or friends. Yep. Yeah.
also recording it.
Mr. Ali, the degree committee here present has assessed the quality of your thesis and your defense, and it took a little bit longer than normally. This is not because we were hesitating about the quality of your work, but we had to discuss the criteria from our university and also from the University of Luxembourg. So we had, in fact, two procedures, administrative procedures, and therefore it took a little bit longer. But I can tell you, in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant upon you the degree of doctor. Professor Kleinjans is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Muhammad Ali, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university. So, dear Dr. Muhammad Ali, I'm pleased to be the first one to be able to congratulate you, but I hand over immediately to the real guys who have been supervising you along these years. Professor Del Sol.
So, thank you very much for your presentation, for all these years of work, and I congratulate you for your degree. Finally, we are here. I have to say, I would like to say a few words about Muhammad Ali. Actually, I was very pleased to work with him over these years, and I have seen your progress, how much you learn, especially from our side, which is more the computational part, and how a good team player you were with when collaborating with other of our team members, and also in collaboration with other partners, especially experimentalists. I think I'm very glad that you're here today and that you finally will accomplish this. Thank you very much.
Thank you very much.
Yeah, Mohammed. There's a serious delegation from Saudi Arabia waiting in the corridor to present and defend next. So for the sake of time, I have to be a bit shorter than I usually am, but I will give you the full version afterwards, okay. Because otherwise it would take too long. Yeah, what's the name, Muhammad Ali? You brought a statement about the boxer yourself in your thesis, so don't blame me for the following few statements now. So there's quite a few in there, but again, I have to skip a few. So maybe the most famous statement of Cassius Clay, so Muhammad Ali, who unfortunately died way too early due to, amongst others, Parkinson's disease. Float like a butterfly and sting like a bee. Butterfly, check. Let's see how these statements relate to you. You're a bit shy, humble, quiet, driven. You're committed, polite, maybe sometimes too polite. Extremely nice, and you've got that particular smile that I see now as well that can make someone's day, at least mine. Float like a butterfly, sting like a bee. I would say remain as you are, gentle in your daily actions and being, and remain soft to those who are around you, to those you care about. But don't be afraid to strike when the time calls for it. I think with your current training, but also whatever comes next, you will soon learn how to sting like a bee. Because in science, that occasionally is necessary. And that brings me to my perspective, a look into the future. Cassius Clay said, I shook up the world, me. And the world is a better place for it, I believe. And we have to be honest at this stage, you, us, we did not yet shake up the world. But it's there, the potential is there. The world is at your feet. It is possible. You have the potential to cure the disease that Cassius Clay actually died of. You can do this. You've got incredible talents. You have worked on your weaknesses over the past few years, and you have grown tremendously. And there's still a lot to be gained. Big data are the future. You are the future. And what more do you want in that respect? And the other PhD students should not listen now to what I'm about to say. We might not really need new wet lab experiments. Because I think the truth is already out there with the generator and the loads of generator big data that are out there already. We are simply lost by all the cues out there. And it's up to you with your current training and data science and computational modeling to unravel it. You have the power to identify and find needles in a haystack. Find those needles and use them to sting like a bee. Let me end by saying that you can be incredibly proud, and I think there's a live stream with your family. They can also be incredibly proud of what you achieve. Congratulations. Enjoy today, change tomorrow, shake up the world. You.
Thank you very much, Daniel.
Dear Dr. Ali, on behalf of the Board of Deans of our university, I would like to congratulate you with the highest academic distinction our university can forgive. And this is the end of this ceremony. I don't know whether there is a photograph to make a photo on the steps, otherwise we go directly to the reception. Thank you very much. I end this ceremony now.
Thank you very much.